I'm going to particularly enjoy this because Jeanette's, as with all inaugurals, provided a bit of a bio. It's on the short side because that reflects, I think, Jeanette's modesty. So I'm going to actually try and make this a little bit more effusive, just to embarrass Jeanette, really. Because I actually work with Jeanette very closely because she's the college director of research. She's part of the senior team in the college. We spend a lot of time talking about not this stuff. So we don't talk about environmental toxicology, etc., microplastics. And as some colleagues will know, in my spare time, I am notionally an environmental lawyer, and actually we would be more interested actually to talk about some of this stuff rather than some of the stuff like REF that we spend quite a lot of time talking about. So here's the formal biography, but I will let Jeanette fill in any of the gaps in the Q&A that you want to. So Jeanette is a professor of environmental toxicology, having spent her early years mostly doing sport. Which sports, Jeanette? Swimming. Jeanette went against family tradition of leaving school at 16 to stay on to do A-levels and further study. She did a degree in biological sciences at the then Napier College in Edinburgh. She says here she became a proud Polytechnic product with further degrees in marine sciences and a postdoc term moving around from Heriot Watt, Glasgow Cali, as she's put here, and west of England. Her projects involved fisheries regulators at each stage and reflects her applied research interests. By quirk rather than planning, which sounds interesting, she then undertook a further postdoc position at Johns Hopkins University, looking at human eye cancer through a model using fish. This was to be the start of mixing environmental and medical research interests, which is actually really useful as we move forward with a range of interests in the College of Health and Science here at Lincoln. Her first lectureship was at the University of Sussex in environmental biochemistry, where she collaborated with nearby Transmanche region colleagues. Together they captured multi-millions of EU Interreg funding to investigate the impact of sewage pollution in the regional coastal area, which has only become more contemporary and relevant when you think about the news over the last couple of years. Having arrived at the University of Hull as a professor, she then moved on from sewage in mollusks to plastics in people. She was attracted to Lincoln by the bright lights of the superior spectroscopy, you put this in purposely, didn't you, equipment, and a research environment where the environmental and medical sciences has all come together in one college. Ladies and gentlemen, I give you Professor Jeanette Rotchell.
Thank you for the introduction. And now you're all thinking of me in wellies, aren't you? Which is normally how I give my talks. So what I thought I would talk about today is mainly sort of about scientific research and a little bit about myself. But before I even start, I wanted to begin with acknowledgements. So I don't actually do any of this work. I've done very little of it myself, and it's all through collaboration, either with other professors, academics, PhD students, undergraduates and postgraduate students as well. So I'm sort of presenting research and the work of lots and lots of different people. So I thought it was important to get that acknowledgement there first. So to start, obviously, I sound a bit odd because I'm going to be talking about plastics in humans and people. I can't call them people because I'm used to thinking of animals, so I call them humans. And I'm not actually from a medical background. I'm an environmental toxicologist. I'm interested in the environment and in particular chemicals in the environment and the effect that they have on the organisms in the environment. So basically, I study poisons or anything that might have an adverse effect. And actually, if you think about it, anything could be a poison. So even water could be a poison if you get it in your lungs, for example. And it's a very interesting career to do environmental toxicology because the chemists very obligingly keep inventing new chemicals. And so I have new things to study and it just never ends. So it's actually a really good career if you are thinking about a career in science. So what I'm going to do today, I'm going to talk a little bit about my journey and keep that very brief because that's the kind of cringeworthy part, I think. And then I'm going to dive into some work around microplastics. What are they? Why am I interested in them? Where do we find them in our diet? Where do we find them in our environment? And then where do we find them in our bodies as well? So I'm going to make this very personal today, and you'll all be thinking about your own body, hopefully. And then I'm going to, because you might be a little bit depressed at that point, I'm going to talk about some things what you can maybe do about perhaps your exposure to microplastics, and things you might be able to do to reduce the levels in the environment and these types of things. So you can go away again feeling a little bit more positive after I've brought you down. So that's what we're going to try to do today. So the obligatory child photograph. So as Duncan said, I was more interested in sport than being academic at school. So that's me in the netball team. And that's also me with David Wilkie, who I thought was quite inappropriately touching my shoulder at the time, but I don't think we worried about that in the 1970s. So I basically spent all of my time doing sport, any sport. I didn't care. I was in every competition, every team that there was. I didn't really want to think about anything academic. And I think as a result of that, I didn't do particularly well in A-levels. So I actually I did really well in history A-level. And on the back of that, I got into Napier College to do a biology degree. And I think what's really weird is I think these days that wouldn't happen. But for whatever reason, Napier saw something in me and were happy to take me with, totalitarian dictatorships in Europe and Tudor and Stuart England. So that was my beginning of an introduction to the sciences, was history A-level. So you don't have to read all of this unless you've got really fast at reading. But in terms of my academic background, I've been all over the place and there was actually no plan. That's the important take-home message here, is I had no idea, no clue what I was doing. And I just kept doing what seemed like something I might enjoy at the next step. So I started in a general sort of biology degree, then I did a marine master's degree. And the reason I did the marine master's degree was because there were no jobs. So I graduated and it was Margaret Thatcher and there were no jobs. So I thought, okay, I enjoyed doing the marine biology, I'll do some more of that. And then I finished my marine sciences degree and there were still no jobs. So I did a PhD. And then there were still no jobs, so I did a postdoc. So there's actually no plan here at all. I hope you're not looking for, you set out to be a professor in this, because that's totally not what happened at all. And I think also the most important part about my background is the reason I've highlighted this fisheries vessel, the Fourth Ranger. So for me, this was the turning point in my life. So this poor PhD student at Napier got saddled with me as an undergraduate project student. They had to take me out with them to do the sampling for their project. And this PhD student, Roger, was looking at the effects of oil in flatfish in the Forth Estuary. And he was using enzyme assays. So he wanted to see, were these oils in the estuary toxic to these fish? And I was going along as an undergraduate project student because he'd been told to take me by his supervisor. So he didn't really want me there. This is the beginning of me and wellies, which carries on as a theme. And I really enjoyed that because he took me out when we went to collect the fish from the Forth Estuary. And then we came back and did quite dull environmental biochemistry laboratory experiments in which we were just optimizing enzyme assays, which is just very repetitive pipetting and measuring. But do you know what? I loved it. I absolutely loved it. I liked the fact that we were doing something that the Environment Protection Agency wanted to do that. It was important to know that. I cared about the fish. I wanted the fish to be okay. And it didn't matter that it was really boring lab work. I just really enjoyed it. And that essentially is where my entire career, if I had one, began, as an honours project student as an undergraduate. And I can never stress enough the importance of making sure your undergraduate project students, when they're with you, have a good experience of their project, because it might launch their entire career afterwards, or entirely put them off science if it goes the other way. So I've been all over the place, and I said there's no plan, there's no map. It was just very random, which I think is okay. So let's get on to the topic of microplastics then. So first of all, why have I decided to talk about microplastics? You've probably seen them in the media. They are quite a topical topic, a fashionable subject, if you like, in the media. Basically, microplastics are just bits of bigger plastic that have broken down. Usually they're broken down in the environment. They've come from like bottles, paper, polystyrene, paper bag, plastic bags, these things. So that basically is what they are. Now, would you be surprised to hear that scientists who research in this field can't agree with how to define them? It's very sciencey, isn't it, that people can't even agree on a definition of what it is that they're actually studying. So what I've got here are just all the different studies that were done up until about 2019, where they're looking at microplastics. And every single study couldn't even agree on what the size range was for a microplastic. So we'll just agree that they're small, okay? They're really small, but they're not very, very small. So they're really small, and if you think of a sesame seed, about 2 millimeters, that's the biggest size for a microplastic. You can see it just about. Then in the smallest size, you'd need a microscope. You can't see it with your eye, so they're teeny-weeny ones. If you want to see them in real life, I don't know if this is going to work. If I go like that, around me, you might see little fibres coming off me, or you can do it with your own jumper. And if you really, really want to, you can get a torch and do it with your jumper when you get home. But you can see them coming off. Those are microplastics, little fibres, little fragments that are coming off your clothing. So the scientists can't agree. They can't agree what size they are. They can't even agree how to classify them. So yes, they're made of plastic. But does that include all of the different plastics? Because actually there are lots of different types of plastics. Some of them are synthetic plastics, some of them are semi-synthetic plastics. They can't agree about that. They also can't agree about how we're going to measure them. What techniques are we going to use? You could use this technique, and it's really turned into, in this field, a bit of an argument. It's a bit like the VHS and Betamax videos. I don't know if you remember that. So, these people thought this method was superior against this product's more superior. It's the same in our research field. We've got different methods, and everybody thinks that their method is the one that you should be using. So there's a lot of debate in this field. How do we get around that? Bearing in mind it's not standardized, and it's important we do delve into this, is to use things to make it more robust, to make these studies more robust. So I'm going to talk about things like blanks. So you might be analysing something, but you could be looking at something here that should have nothing in it. And the reason we do that is because they're floating around in the air, these microplastics. And you might be doing some experiments, but they're all dropping in your bowl while you're trying to do it. So blanks become important. And then there's other things that we need to do as well to make sure it's robust. And what I want you to do when you see anything in the news, anything in a newspaper, especially the Daily Mail, I want you to read it, see where the original study is, and then go and have a look at it and see, did they do blanks? Because if they didn't do any blanks, it means absolutely nothing, because they're probably looking at all of the little particles in the air. So how do we actually do this? This looks very technical. If I give this machine's got a name, she's called Nikki the Nicolette. The guys in the bridge, I think, aren't that keen on me naming all the equipment, but it's going to happen. So you can, as I said, you can see microplastics with an ordinary microscope. But the problem is, with an ordinary microscope, you just see a tiny little fragment. And if you actually were to analyse it using chemistry techniques, it could be something like a piece of wood or a piece of cotton. It could be what we call cellulose, or it could come from cornstarch. That's not plastic. It might even have colour, but it's still not plastic. So it's not enough just to use a microscope. We have to use these special techniques, which is really just a fancy microscope that incorporates a laser that gives you a spectra, and you compare it to other spectra. I can hear all the chemists crying now on how this technique actually works. But trust me, it gives you a plastic polymer type. So it might be something like polyethylene terephthalate. I'm sure you've seen all these recycling things in your bottles. These are the kinds of chemicals I'm talking about. So the first time we had a go at this was using seafood or organisms that live in the marine environment. And actually, this was research that I began because of my colleague, Professor Hua Hong He, who's sitting there. Give them a wave, Hua Hong. So we started doing this research more than a decade ago, and he's from East China Normal University, and it was him that convinced me to get into this research, and we shared a PhD student, Gianna, and we started doing this research here. Now, Gianna and I drove around the UK collecting mussels from different beaches, different rocky shores, and we also went to all the supermarkets around Hull. And we looked at the amount of microplastics in the mussels from those locations. And basically, as a take-home message, we found about 100 particles of microplastic in the equivalent of a bowl of mussels that you might eat for your dinner. So that's 100 little bits of plastic that you were eating every time you eat some of these mussels from one of the supermarkets. And that was quite interesting research. I remember the media headlines, "You're eating your own underpants," I think was the Daily Mail effort on that one. So that was the first study that we did. But I think what was special about that study as well was that we included the chemical characterization. So instead of just looking at them down a microscope, we got really technical. And we really went into the detail. And we included these blanks. We did it in a really, really robust way. And actually, I think this work was, it coincided, not by planning, just by chance, with World Ocean Day, I think it was, in 2018. And David Attenborough quoted it on the news at 6 o'clock that day as an example of research. And I thought, that's it, I'm just going to retire now. He's mentioned my research. That's as good as it's ever going to get. So that was really, really nice. What did we find out then about these mussels? So I've told you about in a bowl of moules marinés, you might have about 100 pieces. So then we went and looked at other fish and shellfish as well. We looked at what you would call premium fish, so ones that are the flat fish that are quite expensive, that you'll get at the fish counter. And we also looked at scallops, king scallops, which are also kind of like a premium food as well. And lo and behold, you also find microplastics in these types of products as well. The fish were interesting in that you only found it in their gills and their stomach. We didn't find any in the fleshy, I was going to point to my stomach, the fleshy bits, the muscles of the meat of those fish. And then the scallops, we found quite a lot again. And I think what it is that scallops and mussels filter a lot of water to feed. So they're kind of like sponges. They're sponging everything out of the water around them to feed. And in doing so, they're also sponging and taking up any little particles as well. So that was our introduction to microplastic research. This is where it starts to get a little bit personal for you. So I had a new PhD student, Evan, and he did a series of systematic reviews. Some of the colleagues in here are probably like, she does do systematic reviews, because I spend a lot of time talking about REF, and you can't put these into these exercises. But anyway, we did do a lot of systematic reviews. And what Evan was looking at was how much microplastic would you find in these common things that people eat or drink. And we looked at salt, different seafood studies, and also drinking water studies. And we converted what we found from this systematic review into what would be a typical kind of exposure, how much are you eating in in your sort of annual daily life. And we found these numbers actually quite large. So if you eat seafood regularly, you're going to be ingesting more than 50,000 microplastic particles per year. If you're having salt, the ground salt that you have to put in a mill, then there's another 6,000 that you're probably adding to your food, to your intake every year. But the real shocker for me was bottled water. So there you're introducing, if you're using just plastic bottled water, more than 3.5 million microplastic little bits into your body every year, which is quite a lot. It's not quite so bad for drinking water. So here I'm talking about tap water. There's less in tap water, but there's still quite a lot in coming out of our tap water as well. In fact, in Actually, I'm not sure this is going to be the case now we have Trump. But in California, they're the first water board to introduce microplastic testing at their water facilities, where you have to have a low level of microplastics, or you can't, there has to be some sort of intervention and filtration before it's allowed to go out to be drunk by people in that area of California. But I'm imagining that's being dismantled as we speak, which is a shame. So we're all ingesting this in our diet. Now I'm going to talk about the work from Lauren, who is another PhD student, and she was interested in what's in the air, what are we inhaling? So we did one study where we found volunteers from 20 houses, 20 homes in Hull and the surrounding area. We had to do the surrounding area because I don't live in Hull and I wanted to do my house, which is in East Riding, very carefully chosen area. And the candy canes at the top here, these candy canes, show you the amount of microplastics that we found in these people's living rooms. So the way we sampled this was we used a very technical method of putting a glass beaker in everybody's sitting rooms, and we waited for the dust to fall in. And this represented 1 month's worth of dust for each home over several months, over a year. And you can see from the different lengths of the candy canes that different homes have different levels of microplastic in the dust in their homes. So we thought, well, that's really interesting. We tried to do a survey as well to think, well, is it because they have different types of furnishings? So we had these really long questionnaires where we were trying to work out what people's homes looked like. Did they have carpets? Did they have pets? Did they have children? All this kind of thing. And when we did the statistics, nothing came through as being really quite strong as a source of the microplastics. But it was an interesting study. So when you go home tonight and look around your room, if the sun's coming in, well, maybe we have to wait a couple of months, you might see things in the air just sort of sparkling. That's little particles in the air, and many of those are going to be microplastics. So then we thought, okay, we'll do it outside. Maybe, it's a bit depressing. We're all sitting in our sitting rooms watching telly in a bath of microplastics. Let's go outside and get some fresh air. So we went out and looked at several different sites outdoors. It was in the city of Hull. I don't think Hull is any different to anywhere else. It's not like you can just not go to Hull and avoid them. And we found at different places we were suburban, right in the city centre, in the city centre next to a trunk road, out by a park, some on the outside. You did find different levels of microplastics, but the microplastics in the air were still present everywhere. So you couldn't avoid them outdoors either. Types of things we were finding, one of them is called pan. So I don't know if any of you have got jackets today that have this kind of puffer type jackets, which feel like they're a bit spongy inside. Yeah, I can see one over there. So that's pan, that's the main type that we found outdoors. PE, that's polyethylene. You get the jumper I'm wearing is probably something like polyethylene, polypropylene copolymer, that kind of thing. That's where they might come from. And some other things, and also some rubbers. Stuart told me not to go into rubbers with too much detail. I'm not sure why, but we won't do any rubber jokes. But I think the rubber maybe comes from car tyres, and these kinds of sources. What can we say about them other than what their polymer types are? Well, they're mostly fibres indoors, but you get more fragments maybe outside. So to me, fibres suggest things like textiles, clothing, curtains, settees, carpets. These kinds of things are the likely sources of those types of things. So that's in our environment. I've talked about what we're inhaling, what we're eating. So now I'm going to talk about microplastics we have in our bodies. So remember, I'm a fish person. So this work all started, the fish stuff started with Hwahong, but the patients, the human studies, started by having a chat in a corridor with a colleague, Laura, who was a biomedical scientist, and she worked on cough, lung and cough. And she was wondering, are the microplastics in the air exacerbating coughs? Could they be, for some reason, causing people to cough? And we were thinking specifically about chronic obstructive pulmonary disorder, COPD. That's what she was mainly interested in. So she was like, Jeanette, can you analyse some lung tissues? I was like, oh, fish don't have lungs. And she was like, it's okay, we can get the lung tissue for you. And I was like, okay, as long as you're going to do that. And we went along to Castle Hill Hospital, and I thought I was going to meet the surgeons, but actually I was in scrubs and in surgery watching somebody having an operation, which is a bit real for me, I have to say, because I usually just see fish or mussels. I don't usually see people going through surgery. So that's how we came to do the lung work, and that led to veins, urine, and blood. So I'm going to give you a little bit of an overview of those. So, sorry, this is a bit depressing. So the long story short is, in the lungs, you can divide it into 3 areas, the top, the middle, and the bottom. And we actually found microplastics in all areas of the lungs. And this is interesting because the lung specialist said there's no way something the size of a sesame seed is ever going to get to the bottom of your lung. It's physically impossible. And we're like, but they're there. So we had a terrible job trying to publish this because they told us it was impossible. And so I guess this is slightly paradigm changing. I do wonder if it's because there may be fibres and they're going down like a spear, like this. It might be very small in this dimension, but very long. And they go down like a spear through the lung. But that doesn't really hold out because we also found fragments of microplastics in the bottom of the lungs. So there is a caveat, however. Out of these people who donated these lung tissues, I think they're all still alive and somewhere in the region of Hull, they found about 40 different microplastics, but these people were all having an operation, obviously. So these are not people who are well. So maybe you could say that only people who are not well have microplastics in their lungs. We're not going to do any experiments on people who are healthy and take out a bit of their lung. That's just never going to happen. What people have done since, though, because of this research, and I guess because we were challenging that paradigm, is there's something that you can do that sounds very unpleasant to me. It sounds like waterboarding. And apparently you put some fluid down people's lungs and you make them cough. And then you collect that. And they've done those kinds of studies, and they found microplastics in that stuff that they coughed back up from their lungs. So there are other studies that have found microplastics there. We haven't completely made this up. Types of things that we find, there's that pan again from puffer jackets, polyethylene terephthalate from bottles, from plastic bags. The other one that's interesting is this thing called PTFE. Which probably nobody's heard of, but has everybody heard of Teflon? Teflon is the commercial name for PTFE. So it's used as a non-stick coating. It's also used as a fire retardant. So it's sprayed on your sofas, on quite a lot of your textiles. I think they've even put it on clothing items as well. So there's a lot of PTFE in your life. It's also called one of these forever chemicals, which means it doesn't break down, which is a problem. Plastics generally don't break down. They're around for hundreds and hundreds of years, and they have other effects as well that can disrupt your endocrine system. So quite serious chemicals that we found in the lung. So that's the lung. Are you getting depressed? Yeah. So then we did a study using blood from 20 volunteers. These were people that came into the University of Hull. So they were mostly academics, people from professional support services. I would hope they were all healthy because they were able to walk in and give us a blood sample. And what we found here, again, cutting a long story short, is that we found 18 out of 20 donors had microplastics in their blood. So if you look around you, 1 out of 10 of you would not have the microplastics based on that. There was also a Dutch study at the same time that's been published, and they looked at about 20-odd people, and it was the same sort of 80-90% of people had microplastics that they detected in their blood. So whatever those people were doing that had none in their blood, well done. I want to know what your home looks like and what you're eating. Or it might just be because we were only analysing a small amount of blood that we caught you on a day when that bit of blood didn't have any microplastic in it. We just don't know. So there have only been literally 3 studies worldwide so far. Our study, the Dutch study, and they've repeated it with another 60 donors and found the same sort of results, and another study in China that's just come out. And what's interesting is that the community is desperate for this not to be right, for this not to be true. There's all sorts of things wrong with our methods, is what we're hearing. With our methods, it was something to do with our procedural controls weren't good enough, but actually they were as good as you can get. With the Dutch study, they were saying your gas pyrolysis method is rubbish. And we're not sure where this is coming from, whether this is coming from big business or industry, or what this is about. But it's kind of interesting that we're getting that pushback on these results. We got that from the lung work as well, but to a lesser extent. Urine, I have all the fun jobs. So here, these are ladies who had endometriosis, and we wondered if endometriosis is an inflammation type problem. Maybe little bits of plastic are causing inflammation, and that's why they have, or that's one of the reasons why they have endometriosis. We didn't know. So we analysed the wee from some ladies who have endometriosis and some healthy volunteers who gave us their wee as well. And again, we found microplastics in their wee. We found more in the endometriosis sufferers, but not statistically more. So we couldn't really say, we're only using a small cohort of women here. We did wonder, it got very surreal at one point. So usually I'm sampling with fish out in the environment or mussels. But here we seem to be getting a lot of fibres from clothing. So we actually had to write a protocol on how to do a wee so that we could get a really good sample without clothes. We called it fluff. We had to write it in a way that wasn't too scary. So we had this whole protocol of how to do a pee, and we still were seeing a lot of fibres that we thought, I think these are coming from people's underwear or the room. So then there are some ladies who were undergoing surgery for endometriosis, and they needed a catheter fitted. So they had a metal catheter fitted, which I felt really bad about, because a plastic catheter is obviously more comfortable, but they were under an operation and anaesthetic, so I'm not sure they really felt the difference. But we thank those ladies for giving us those samples. And in those, we found very, very low levels in those healthy donors, in those samples, very low levels of background microplastics. So it gave us confidence that what we were finding were real in the urine. And the reason we wanted to be really super confident was because, again, it's a bit weird because I'm talking about a human body and not a fish. Anything that gets to the urine is normally going through your kidneys. And your kidneys are like a very big filter. And they've got very, very thin channels that go through them. And in theory, a microplastic, something the size of a sesame seed, is about 1,000 times too big to go through your kidney. It's just not possible. So we're like, oh, this is really weird. If we're finding these in the urine, this, again, is messing up the entire paradigm of what can pass through a kidney. So we're desperate to find another reason for how it got to the bladder. So actually, there are blood vessels as well that go around your bladder. And apparently, they could have come through the blood vessels. I just don't know. But since then, there have been other studies that have found microplastics in the kidney as well. So we're not the only people to say, maybe we need to think about what can pass through different parts of the body. So if you're going to be an optimist, you could say, well, okay, I'm eating all this food and I'm inhaling it, and it's ending up in my body somewhere. It's fine, I'm just going to wee it out and I'm going to poop it out. It's gone. It's not staying in my body. It's not going to be doing any damage. So that was the next question. What does that have on? What impact does that have on us? How does that affect our health? That's the big question, isn't it? Is this something we need to be worried about or not? So Evan did another systematic review, this time a meta-analysis as well. And we looked at all of the toxicity studies that have been done to date by all the scientists around the world. So these aren't obviously, we're not feeding microplastics to people. We're usually using small samples of cells in a Petri dish or in a test tube, or you're using maybe animal models, these kinds of approaches. So it's not always the same as using human subjects. And what Evan's done here was he looked at all the different cell studies, so these are very artificial, and he brought all the evidence together, and he identified some of these responses in the cells that were definitely triggered by the presence of microplastics. So cytotoxicity is cell death. The cells just curled up and died when you expose them to these microplastic particles. Immune response speaks for itself. So there are markers of the immune response that people measured. And again, those were affected by the presence of these microplastics. Oxidative stress, this is something that also leads to problems in cells. And then barrier integrity, this is where things are maybe crossing over from one cell to other cells and transporting around the body and interfering with that potentially. So the answer is that in a lab environment, very artificial, using cells, these do have some biological impacts, and perhaps they have health impacts as a result. Now, the disclaimer here is that all of the toxicology experiments, and this is where I want to hold my head in my hands and cry, they tend to use round microplastics to do their tests. And you'll have noticed up until now, I have not mentioned a single sphere or round microplastic. We've never found them in a sample in the body, in the air, or in the muscles. We find fragments and we find fibers. We don't find spheres ever. But they're doing all their toxicology tests with spheres. Now, if you imagine trying to swallow whole a grape, it's going to go down, isn't it? It's not going to hurt too much. If you try to swallow a piece of Lego with hard edges, that's going to hurt, isn't it? You've got more of a physical presence because of hard edges. And it's the same with fragments and fibers. So these guys, great, thank you for doing all that work. Can you do it all again now with realistic microplastics that have hard edges, please? So there's that. The other issue that I've not even mentioned, the elephant in the room, is that you're probably thinking plastics are just made of this or that, one chemical. They're made of, I don't know, nylon or polyethylene. Well, actually, plastics contain up to 20,000 different chemical additives. So when you look at a piece of plastic, the manufacturer just tells you what the plastic polymer is. They don't tell you about the other 20,000 things that they might have put in there that changes their qualities and makes them more bendable, it makes them more resistant to fire. And some of the, well, about 2,000 of those additives are known carcinogens or endocrine disrupting chemicals. And at the moment, they don't have to state any of that on their products. So in addition to the fact that you've got this physical presence in your body, you've also got a pool of plastic that are leaching chemicals that are toxic as well into your body. And that's something you need to think about as well. They're also, how should we call them? They're vectoring. It's like they're taking a ride on a bus. So you've got the plastic particle, but sitting on them might be bacteria or other chemicals that have decided to associate with those as well. So they're like a bus of lots of other toxic stuff coming along with them as well. And we haven't really been studying those yet. So it's a much bigger sort of question here than we think. So I'm going to talk a little bit very quickly about surgery. I was very taken with my experience in the operating theatre. And in particular, once I got past the fact that there was somebody being operated on, I looked in the room and there were about 8 to 10 people in there. Because you never know this, do you? Because you're always asleep when you go into an operation. You don't really see what's happening. There's all these people in the room, lots of movement, but the amount of machinery is extraordinary. Really, really a lot of machinery. So that got us thinking, because guess what all the machinery is made out of? Plastic. So here you have someone who's really very ill, wired up to a whole lot of plastic. And in working with the colleagues from Castle Hill Hospital, we decided to look at a heart bypass operation. That's what they specialise in. And basically, if you're in a place where you need a heart bypass operation, you're obviously not well. But while you're in there being operated on, a machine takes over the function of your heart so that they can do the bypass operation. And I brought one with me, not heart, because that would just be weird. So this is what keeps you alive while you're being operated on. And that operation will take about 3 hours. And that's what's going around. And that's what this diagram is trying to show here, are all these bits inside this heart bypass machinery. And the interesting bit is this would obviously wires going to different bits and bobs. This helps to pump the blood around, and there are pumps that squeeze like this. And it's this squeezing, engineers have got a name for it. Spallation happens. Things shed, basically, bits of plastic, it's all plastic, can shed while it's being squeezed for 3 hours. So we thought, I wonder how much plastic is introduced into a body when you're on one of these. And we worked that out, and the answer was, depending on the type of machinery, if it's one of these, a conventional, this is conventional, there's about 60 microplastic particles introduced every hour while you're hooked up to this. If you're on the little ones, a mini one, which was actually quite shocking, it looked to me like a little plastic bag with not a lot else going on. So that was a bit sort of overwhelming to think, gosh, that keeps somebody alive. So then we thought, okay, they're introducing plastics into somebody's body who's already very ill. And I've already been talking about immune response, inflammation, oxidative stress. What's that going to do to somebody who's recovering from an operation where you've just introduced all of those into that person? That's for the surgeons to worry about. But then we got into the types of polymers again. So the names of the plastics are different here. And we're like, well, they don't tell you what it's made out of. It's not like it's got a little sign here. It's like, oh my goodness me, what are we going to do now? So we took a hammer to it, and then we just started to look at all the different parts. And then we'll go to the next bit. That was what we were doing with the equipment that they have at the bridge. So they have a handheld Raman analyzer. So you can just point it. It's what they use at airports. You can point it and it tells you what it's made out of. So we're able to do that for the entire piece of equipment. And we were able to work out, okay, so these things, these are producing quite a lot of the plastic particles, which makes sense because it's being pumped. And we were able to then look at what goes with what and work that out. So the next thing is, well, what are we going to do about that? That's not great. So during this, a brief interlude, moved from Hull to Lincoln. And as I said, I was attracted by the facilities at the bridge. I've been using this micro FTIR, so I was like the Betamax, and there were these other people using Raman. They're like the VHS, and we have the Raman here at the bridge. I was like, I can come here and I can do all my analysis using both techniques, and that will stop any issues of the debate of you're not using the right techniques. And then we got into more details. We're carrying on this. We're still in the surgery, in the operating theatre. We thought, oh, let's just fill our boots now. Let's just see what they've got in an operating theatre. So we've got drapes, plastic. Don't know what that is, but it's wrapped plastic. It's wrapped in plastic. Everything's in blister packs. That looks like it's got something to do with collecting urine, but I'm told it goes down your throat like this when you're on respiratory system. Plastic, all bits of things, basically all plastic. You get the picture. Everything in the operating room was made pretty much from plastic, or it was wrapped in plastic. And the first thing they do when they go in there is open all the plastic. There's plastic everywhere. So we got a small amount of money to look at 8 different bits and bobs like this. And we found, I can't remember, it's about 26 different types of plastic in all these different pieces. That's what this pie chart shows. So lots and lots of different types. Some of them we've already seen before, like polyethylene, polyethylene terephthalate, nylon, and some other ones that we hadn't seen before, as well. So plastic pretty much everywhere. So what are we going to do about this? So we were working with the surgeons to see, well, what can we replace? What can we get rid of in that environment to make a difference? And so we did this, probably the weirdest experiment I've ever done in my life, where the surgeons got medical students to come along and to do an operation on one day. There wasn't a body, it was just a fun, like, mock operation. And we spent the day doing a pretend operation. And then the next day we came back, we took all the plastic out of the room to see if the dust changed. And I can't even tell you what the results are because we're still analysing it. And the people that are analysing it are sitting just here at the front as well, no pressure on those guys. So what we're thinking about is what can we replace? Because, you know, we want to be able to do something about this. What else can we do to make things change? So it can be a bit depressing. So I've written verbal evidence, or written evidence rather, for Parliament about the air work and what we're breathing in and what we're finding in our lungs. So I'm trying to influence policy. I'm also trying to influence the regulators when I do that. I've also been on the Food Standards Agency and introduced the microplastic research there in the hope that they might introduce some regulation. And I've also been involved in a documentary. So you can't, I don't think it's been released yet in the UK. That was a really interesting experience. So when I talked about the blood work, one of the donors was the presenter of that documentary. And she has her poop analysed, her blood analysed, and they talk about the results of that. And then they also talk about big industry and chemicals and all the rest of it as well. So I'm doing everything I can to try to influence plastic and the use of plastic. But more importantly, what can you do? Okay, this is where it gets super personal now. I'm going to try and do a video and see how it goes. I didn't think you'd want to see me in Lycra, so this is why we've got this video.
Hey gang, it's 2023 and it's time to look after yourself and the rest of the world. Today we're going to work out how you can work microplastics out of your everyday. So what are microplastics? Well, they're tiny plastic particles that break down from larger plastics in the environment. Are you ready? Let's go. Let's do the air dry. Did you know that a single tumble dryer releases about 120 million microplastic fibers into the air each year? You know what the answer is? Get the air dryer out and save the environment. Next up is the Hoover maneuver. 40% of all dust particles come from, you guessed it, microplastics. Now, there's only one answer. Get them out the door. Do it. You hear me? Say yeah. Ditch the plastic and get some class with your glass. Plastic bottled water contains 50% more microplastics, so choose glass. It's a completely non-toxic natural material. Next up, the wave. Less microwave responsibly. No more plastic in the microwave. When plastic is heated, microplastics can get into your food. So there's only one thing to do. Wave them away. Let's do the sip and repeat. Beer mug. Don't use paper cups. Takeaway cups release trillions of microplastics when exposed to hot liquids. So come on, think about it. 5 top tips, 5 jazzy exercises. In 2023, let's work out what microplastics are, how we can produce less, and feel good.
So hopefully you enjoyed that, and it was better with him doing it and not me. So that was an interesting collaboration, actually. So I do a lot of consultancy as well as research. And on that project, I was working with the glass industry, and they wanted the background, the stats, the information for Mr. Motivator to get his point across. So I had to do a whole lot of radio interviews with him, which was great because I got to meet him, but he got the point across way better than I ever would, which was excellent. So you've got some things to do. I also wanted to mention your frying pans because of the Teflon and the PTFE. So if you've got really old non-stick pans and they look a bit shoddy, I would get rid of those and just use not non-stick. just use, a metal type frying pan as well, because little bits come off over time and they end up in your food as well. And that pretty much is it for my talk. I hope you found it interesting, and I hope I've given you something to think about, and you're going to go away and do your Mr. Motivator from now on. And I'm happy to take any questions. You've got to remember that we didn't know the Dutch people were also doing their study, so we were the world first, and we didn't know what we were going to find. So we thought we'd do a pilot study and let's see what happens. And actually, you clearly have never had to apply for ethics. Maybe you have. So the ethics of trying to get these samples, it's very, very convoluted. It can take longer than doing the actual research itself. It's a lot. And then, so that they also say, well, why didn't you get the ages, the professions, the occupations? The ethics would have taken even longer to do that. So I think now that we've shown that it's a proof of principle, we can go back and do much bigger scale analysis. The other issue is that our methods, they give us a lot of information that's very rich about like the shapes and the sizes. Whereas if you use gas pyrolysis, it's very quick, but you get nothing about the shape and the size. And you remember the bit about the grape and the Lego cube? It's really important about the shape. And when I say it's rich data, it's a lot of time sat at that special microscope. You ask these two here how long they've spent entire summers doing this analysis for just 20 samples and the 10 blanks. So it's hugely labor intensive. There's no quick way of doing it. And I would love somebody to give me more money to be able to do more numbers. And can I just also plug that Kat and Charlotte are looking for PhD studentships. And they're very, very good lab researchers. So please, if anybody's got any funding, these two would be very good as PhD students.
Yes. Allow me, thank you for the talk, Rachel. I'm a professor of journalism, so I'm very happy that some journalists are being trained. I've taken interest in what you are doing. Now, I'm interested in two aspects. Number one is the one that you did with the sick people. Now, did you measure what plastic was in their system before they came to the hospital, and then what was added to it?
No, but that's a really good experiment. And it's the one that we've just written the bid for that should be submitted this week to Rose Trees. So the one where we're in the operating theatre was a pilot experiment funded by the Rose Trees charity, and they just gave us about £10,000. But now we're putting in a much bigger bid for £300,000, and that's what we're going to ask. So before somebody has their operation, before they're put on this thing, we get the blood sample before, and then after the operation, we get the blood sample after the operation. So we can then relate it. We also want to get the health outcomes for those people who've had that intervention, that surgery as well. So it's so difficult to get across this and stress that this is such early research, that we're just beginning with it at the minute. We could do so many different things though. Yeah.
So what would be an ideal world for?
We could go back to the 1980s. But seriously, we're basically doing a big experiment on the planet with everybody on it. And I don't know what we can do. I think we need to know quite urgently what the health impacts are. So for instance, does it exacerbate chronic cough? There are some suggestions that it might exacerbate inflammatory bowel disease type health problems. So we need to do all those kinds of studies now. So one thing you could do, because we were able to work out that it was the squeezing of this that was the source of some of the plastic, is to look and see if there's a different plastic polymer that you could use instead there. It might be less flexible, but it might shed less. So those are kinds of things we can do. But I kind of wonder, what did we use before we had these? Can we do a bit of sort of thinking about that? But the other thing that probably scares me the most, and I'm not going to say anything because this is being recorded, is that a lot of big industries have a vested interest in us using plastics. And that's the scary bit, because they want us to use plastics, that it's not in their interest for us to use alternatives. Yes, it does. So the air conditioning in the operating theatre is obviously going at a very, very high rate to keep the operating theatre very sterile. And what was surprising to us was we still found microplastics in the air in that environment. So even if you have very strict air conditioning, you still have microplastics in the air. I don't know if that's because of their weight. I have no idea. But we did take, so we had a mock operation with plastics, a mock operation with no plastics, and then we also did it several days later when nobody's been in the room and the aircon's been switched off. No microplastics in the air. I think they've all fallen out or disappeared or wherever they've gone. So air conditioning is a factor. In your home, it's good to, I think they knew this in Victorian times, open the windows, get some fresh air. Although it depends where you live, because if you live next to an A road, you're going to have a lot coming in off the roads from tyre plastics and things.
Thank you very much, Jeanette. So I'm sure Jeanette would be happy to have a chat with you individually if you want to join us for questions afterwards. But I do have a quick plug for the next Inaugural, which is Professor Anna Tarrant on the 26th of March. And she'll be talking about co-creating a far less inclusive society. So I hope you'll join us then. Thank you very much, everyone, and thank you, Jeanette.